Happy World Carnivorous Plant Day. Firstly, I want to thank ICPS for hosting the World Carnivorous Plant Day 2022. I also want to thank everyone for attending this talk. My name is Sukuan Liu, currently a graduate student from Smith Lab of the University of Colorado Boulder. Today, I will be presenting the research done by me and my advisor, Dr. Stacy Smith, on the evolution of South American March pitcher plant, Heliamphora. We will start by talking about a little bit of the background of the pitcher plant family, Sarraceniaceae. Sarraceniaceae is a carnivorous plant family in the order Ericales. They're related to both the kiwi fruit family, Actinidaceae, and semi-carnivorous plant family, Rorigoaceae. Ancestral Sarraceniaceae was estimated to have split from the lineage giving rise to Actinidaceae and Rorigoaceae at around 88.1 million years ago. Sarraceniaceae is found exclusively in the New World, while all of the Rorigoaceae and most of the Actinidaceae are found in the Old World. Within Sarraceniaceae, there are 3 genera distributed in North and South Americas. One interesting fact about their biogeography is that the closely related genera Sarracenia and Heliamphora are actually distributed in 2 different continents. Sarracenia is native to the eastern US and Canada, but Heliamphora is endemic to the Guiana highland of South America. The least related monotypic species, Darlingtonia californica, is found in small populations in the Pacific Northwest and Sierra Nevada. There are many hypotheses on the origin of Sarraceniaceae, but for today's talk, we will focus on the evolutionary history and the biogeography of Heliamphora. So Heliamphora is the most species diverse genus in Sarraceniaceae. There are 23 described species and many more undescribed taxa. They generally occur in the Pan-Tepuy region or the higher elevation of the Guiana highland. The Pan-Tepuy region is composed of many fragmented and isolated tabletop mountains, and they are called the tepuys. It was thought that those tepuys was formed by ancestral mountain uplift and subsequent erosions. The climate in the Pan-Tepuy is constantly cold and humid. It's drastically different from the warmer and drier climate in the surrounding tropical lowland, thousands of meters below. The majority of Heliamphora species are found to be distributed in their typical habitats, and those are the open clearings, marshes in the upper mountain region, and Tepuy summits, with elevations above 1,500 meters. Some species are found to be distributed in much more extreme microhabitats. Species like Heliamphora hexapenticulata and folliculata are often found to be growing out of crevices of exposed canyon walls on the tepuy tops. 2 species, however, are found to be occurring in much lower elevations. Heliamphora ciliata and heterodoxa are found in small populations in swamp and open clearings in the lowland savanna that is called La Grande Sabana. In the pictures on the right, you can see the environment in La Grande Sabana is a lot different than the Pan-Tepuy region. That's much hotter and drier. As we have seen so far, the distribution of Heliamphora is fragmented and highly endemic. They're usually distributed in a cluster of nearby tepuys. Some species have such a narrow distribution that they're only found in a single tepuy. You can probably visualize the high degree of endemism here in the picture. You can see the fragmented eastern tepuy chain that is about 60 kilometers or 37 miles long. In the just 60 kilometers of stretches across the tepuy chain, you can find 5 endemic Heliamphora species that don't occur anywhere else. The distribution of Helianthera species are generally confined to two major clusters of highland area in the Pentapui region. They're separated by the extensive lowlands region that might be acting as a dispersal barrier. Morphological similarities observed among the western species also suggested that they might be descended from a common ancestor. A cluster analysis on the floristic composition of 40 tepuis has also shown that there are more differences between eastern and western regions, and more similarities within each of the regions. Botanist Basil McGuire and ecologist Otto Huber have also hypothesized that the high degree of endemism in the Pentapui was facilitated by the erosion of the Roraima Plateau. It was thought that the endemic taxa, like the ancestral Helianthera population, used to be distributed continuously throughout the plateau. As erosion gradually occurred, those ancestral Helianthera population became gradually fragmented and eventually diversified through vicariance or allopatrid speciation. If the vicariance hypothesis was true, we should expect geographical closely distributed species to be more closely related. We will also expect sisters relationship between the eastern and western groups. In other words, we expect all eastern species to be more related to each other and vice versa for all western species. In order to test our hypothesis, we need to construct a phylogeny of Helianthera. We sampled 22 out of 23 described species plus 2 undescribed taxa. We ran phylogenetic analysis with 3 nuclear markers using Bayesian inferences and maximum likelihood with some secondary node calibrations. We did ancestral state estimation of some morphological and ecological traits. To test for signal in geographical clustering of related clades, we have run Mentel tests, which test if there is a correlation between genetic and geographical distances. The estimated phylogeny has great support for major clades and subclade. There's also some okay support for relationships within each of the clades. Based on those estimated phylogenetic relationship, we name each clade and subclade by their geographical distribution. Using the estimated phylogeny, we were also able to infer some of the ancestral state of morphological characters. One of the traits of interest is the evolution of drainage structure unique to Helianthera, which is used to drain out excess rainwater while keeping the captured prey inside of the trap. We found the ancestral Helianthera population probably has drainage holes, while E1 clade, including Helianthera minor and ciliata, further modified it into drainage slit. We also looked at the evolution of scape pubescence, or the pubescence of the flowering stalk. We found the pubescent scape was probably ancestral to the genus with multiple transitions into glabrous scapes. Based on the phylogeny and known geographical distribution of Helianthera species, we confirmed that the western species are monophyletic. However, the western clade W is closely related to the eastern clade E1 instead of to all of the other eastern species. In other words, the western clade W is nested within all of the Helianthera. When we ran the Mentel test, the result was significant, showing genetic distance is indeed correlated to geographical distance. In order to make sure that it's not because the western clade that's heavily affecting the results, we reran the analysis to exclude all western taxa, but the results still came out significant. The eastern species, however, has more puzzling distributional pattern. Allopatrid speciation itself alone doesn't seem to be able to explain the more widespread and intermixed patterns. especially when we look at the diversification trend of Helianthera within the last 45 million years. We can see that major clades have already formed by mid to late Miocene, potentially through vicariance from the mountain uplift and subsequent erosion. But by mid-Pliocene, it was thought that the Pentapui region has become more or less similar to today's landscape. Strangely enough, at around the same time, we start to see rapid diversification of Helianthera species occurring within each of the clade. If we look at the trend of global average temperature during that time, the global climate has just entered a phase of thermal reduction and oscillation. At least 40 repeated glacial interglacial cycles had occurred until present. If we just zoom in onto the global climate trend in the last 500,000 years, we can see there were at least 4 repeated glacier and interglacial period. In each of the cycle, there were more than 10 degrees Celsius of thermal shifts, possibly affecting the distribution of thermal temperature-sensitive Pentapui endemics. And this allowed them to migrate downward for more than 1,000 meters to the surrounding lowland. It's very likely that the cold climate adapted Helianthera would have responded to the thermal oscillation through vertical displacement. During the cooling glaciation period, the Pentapui climate would have moved downward to the surrounding lowland and therefore allowing Helianthera to migrate down the elevation. And in the warming interglacial period, similar to what it is today, the Pentapui climate would have moved upward back to the Tepui top, driving cold climate adapted Helianthera to move up in elevation. The populations in the lowland would either go extinct or remain as small populations in microhabitats with a climate similar to those in higher elevation. Present-day lowland species Helianthera ciliata, found in Lagland savanna at the elevation of only 900 meters, could potentially be a glacial remain. In a simulated map of Pentapui climate during the last glacial maximum, we can see the white area representing present-day lowland habitats has been cooled enough to have climate similar to the present-day Pentapui region. This would allow higher elevation endemic like Helianthera to disperse and migrate. In the reconstructions of available migrating space for species with elevation range similar to Helianthera, we can see that the whole eastern Pentapui area would have been virtually continuous for Helianthera species during the last glacial maximum, therefore allowing eastern species to disperse, mix, and hybridize. In conclusion, we found that major clades have likely formed through allopatric speciation with the erosion of ancient landscape. We also found that vertical displacement and local migration during the Pleistocene thermal oscillation have likely contributed to Helianthera's present-day diversity. It's very possible that processes like hybridization have played important roles in Helianthera diversification. However, our study was not suited or designed to understand questions about recent hybridization. Future studies with extensive taxon sampling in the field and genomic sequencing will be needed to answer those questions. I will end the talk with a little bit more pessimistic evolutionary future of Helianthera in the next 100 days. The global temperature is projected to increase 2 to 4 degrees Celsius by the end of the century, hotter than any interglacial period ever experienced during the Pleistocene. Such projected temperature increase will cause Helianthera's suitable habitat to shift upward 300 to 600 meters in elevation. For example, Shimentap Massif, one of the largest and most species diverse Tepui cluster, according to the simulation, will be losing 25 to 57% of suitable habitat for the Pentapui endemics. Heliamphora species restricted to smaller tip-leaf summit with lower elevations would lose suitable habitats because there will be no higher place to migrate to, and they would eventually go extinct. So it's possible that we need conservation effort in preserving species with limited distribution in the warming future. At the end of the talk, I want to acknowledge the many people that helped make this project possible. I want to thank Dr. Andrea Fleischman for sharing data from the pilot study, important taxonomic information, permission for using and modifying diagrams using the paper, and valuable comments for the drafts. I also want to thank Dr. Charles Davis, Dr. Aaron Ellison, and Elena Butler for sharing raw data from previous paper. I also want to thank everyone in the Smith Lab in the eBio department and the IQ Biology program. There's also an amazing lecture by Dr. Stuart McPherson with details of Heliamphora species and habitat on YouTube, and I strongly recommend you guys to check it out. If you want to know more about this study, welcome to contact me with questions or request a copy from me. Thank you.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.